Welcome back to our channel Go for Agriculture. So in today's lecture we're going to see the most important and easy and so interesting topics: pest survey, monitoring, sampling and sampling methods. So let's start with our lecture: pest monitoring. So basically pest monitoring is checking the insect pest or the natural enemies in the environment, and it is the most fundamental tool in the IPM for the management decisions. So this pest monitoring, it provides us some of the information like what kind of pests were infecting the crop, and also what is the density of pest, and if the pest is infecting the crop, what are the control measures to be taken, and if we take the control measures, were they effective, like are they suppressing the pest population or not. So these are the most important information we can get from pest monitoring. Next is how to identify the pest. So we can identify the pest with the help of pest insect remains. So like for example, if there are cockroaches in your home, so you can easily identify them with the help of their remains. And then second is frass. So basically frass is a product of the insect feeding and digestion. So like for example, in your home, if there is old furniture kind of thing like a wooden structures, so there are some insects called as termites. They start feeding on those old wooden things and they convert that into a powdery form. So that powdery form is called as frass. So with the help of that frass we can know, okay, so there are termites which are affecting my wooden thing. Next is visual damage. So let me tell you with an example. It is like, have you ever opened a book which is really so many years old? So when you open that kind of book, you can see some sort of holes or the damage to the papers. So this is because of an insect called silverfish. So this is a visual damage because you can easily identify it. Okay, there is a damage in this book. So this is visual damage. And also you can identify with the help of the smell and sound also, because each and every insect, they produce some sort of foul smell, some of the really foul smells, and also they produce a different kind of sounds. So with the help of smell and sound also you can identify the pest. So these are the different strategies to identify the pest species. Next is sampling. So basically sampling is a repeated systematic data of an organism, like it's collecting the data of an organism in its environment. See for example, in a field, and if you want to collect the sample of the soil, so you would collect the sample of the soil, and of course in the soil there will be different kinds of organisms. Okay, so you'll take them to the lab and you do the testing. When you do the testings, what are the things you would know? They are the what kind of pest is there, and what is its life cycle, and what is its different kinds of host, etc, etc. So with the help of this sampling you can know different systematic data about an organism. And what is sampling universe? So sampling universe is the area which is being sampled. So like for example, if there's a 1 hectare area, so that 1 hectare field is the sampling universe, and the sampling unit is within the sampling universe. If you select a plant to do the sampling, so that plant is called a sampling unit. So it can be a traps or a leaf, or a plant can be a sampling unit. So now we understood between the what is the difference between the sampling universe and the sampling unit. Next is, why do we do the sampling? Okay, so basically we do sampling to know the presence of the pest. So presence is also called as qualitative analysis of the pest, and population of the pest. If you know the presence of the pest, automatically we'll know the population of the pest, means the quantitative. And also we can know the injury, what is the injury cost. If you know the injury, we will know what is the loss caused due to the pest, and also what is the quality loss and the value loss, and also we can know the damage cost. So this is why we do the sampling. So the sampling is helpful to predict the loss production, quality or value, and the damage cost. Next is pest survey. So pest survey basically is an official procedure which is conducted to determine the characteristics of a pest, or to determine the pest species occur in that area. So if we say what are the types of pest, one is a roving survey, and second is a fixed plot survey. So roving survey is the assessment of pest population from randomly selected spots in a large area. So like for example, if this is a huge field and you select some of the randomly selected spots and you collect the samples from those spots, so this is called as roving survey. And the main advantage is you can cover the large area in a short period of time, and also it provides information on the pest level of infestation in a large area. And the next is fixed plot survey. So this is the assessment of a pest population from a fixed plot of region. So like for example, if you take a fixed plot and you collect the samples from this plot from everywhere, so this is the fixed plot survey. So the data on pest population recorded periodic from sowing till harvesting. So this is done in the fixed plot survey. Next is sampling techniques. So basically there are two sampling techniques. One is the absolute sampling and second is the relative sampling. So absolute sampling means it is to count the pest occur in a plot, counting the number of pests we can say. And the main advantages is we can know the population and also we can know the mortality rates. And basically this absolute sampling is used in case of immobile and the large insects like cutworms and caterpillars, because they move slowly and they are large, we can easily count them. But what if the insects are mobile, like if they fly, what we can do is relative sampling. So in case of mobile insects, we do the relative sampling, and it is to measure the pest in terms of values so that they can be compared over time and space. Like at this time and at this region, there are these many number of mobile insects. And the examples were the light traps, pheromone traps, etc. So these are the two sampling techniques. And the main question is how to sample. So in case of this field given, you can, all the people who do the sampling, they basically follow the W or the Z, so the zigzag shaped, like this is a W shaped, so that it covers all the points of the field, so that you can get the accurate results during the pest counting or the pest, in the lab we can say. So this is done for the entire field. So what in case, okay, see like for example, if there's a farmer and he have a field, but he divided the field into two parts, like one part of the field he's cultivating a cotton for example, and the second part of the field he's cultivating rice. So for cotton and rice there will be different management strategies, and also different chemical fertilizers application, and also different kind of pest will be. So what would you do in that case? Let me tell you. This is how you do the sampling in the fields which were managed differently. Like you'll collect the samples in this manner so that it covers all the sides of the field. Okay. Next is sampling characteristics. If you know the characteristics of an insect, you will know how to sample. So the main characteristics of the insect is the aggregated form. Like they'll just be in groups at different places. See in this picture you can see they were in the different groups. If I should tell you in the language you understand, they will be with their best friends in a group at the different spots. So this aggregated form is called as clumped, aggregated or the clumped form. Or these insects, they can be in aggregated form or the random also. See, if they are in the uniform, we can easily collect the samples from the one spot only, so there will be no problem. Since the insects, they were in the aggregated or the random form, we should do the zigzag method so that we can get the accurate results. Next, and also with the help of the life cycle also, we can know where and how to collect the sample. See, like for example, in the overwintering, like when the insects it goes into dominance, we can know when to collect, where to collect, and at what stage to collect. And also we can know what kind of host if they attack, and what kind of plant parts they attack, and what is the damaging stage, and what are the number of generations per year, and what is the generation time. So because of these things, we can know where and how to collect the sample. Let me tell you with an example. If you see the rosy apple aphid life cycle, so this is a rosy apple aphid life cycle. So in this we can see fall, spring, summer, and this is the winter. Okay, So in this you can see in the winter these insects they hatch to wingless females and then gives birth to wingless females and then give birth to winged females, and later they migrate to summer host. This is the most important thing. In spring they migrate to summer host, and again in summer several generations will come, and again the winged females again they migrate to apple. See, for instance, think that you're a farmer and you're having an apple orchard and you want to do the sampling, but you know that your field is affected with the rosy apple aphid, and in which season would you do the sampling? Is it fall or winter or spring or summer? I would recommend you to do the sampling in the fall, because in summer and winter, sorry, in spring and summer they would migrate to their summer host, but in fall and winter they would affect your apple orchard only. So this fall and winter is the best season for you to collect the samples. So the farmer has to be well aware of the characteristics of the insect, so that he should know what are the hosts that the insect affects, and what are the stages of the insect. If the farmer is well aware of the insects, so that he would know at what time we should collect the sample, so that it will give us the good results. Next is sampling methods. So first are the visual samples. The name itself says visual means seeing. So in this visual sampling, with the help of the hand lens, we'll count the insects directly on the damaged leaves or stem or fruit or roots. So in this picture you can see this man is using his hand lens to observe the pest on the infected parts only. So basically this visual sampling is used to count the aphids, scale insects, caterpillars, mite leaf hoppers. Next is sweep net samples. So basically in this a net kind of thing is used to collect the insects. So this sampling method, it collects the insects really quickly, but it is not useful in tree and the small fruits, because you can see how the sweep net is. Can you collect the insects from the fruits or from the trees? No, it's not possible, because this is used to collect the insects from the huge canopy of the crops or from the grasses kind of thing, so that we should have a ground cover. So in this, one sweep is equal to the one pass of the net, and the return sweep you do, it is a second sweep. So for a site the 10 sweeps is okay. So this is a sweep net sampling. Next is vacuum netting. So in this vacuum netting, a strong vacuum pressure is used to suck the insects from the plant canopy. So like you can see in the picture, a woman, she's using a vacuum, so that she's covered the mouth of the vacuum with the net, so that this vacuum pressure it sucks the insects, so the insects get stuck on the cloth. And also on the left hand side you can see the huge machinery is used for the vacuum netting. So this is vacuum netting, and basically this is used for leaf hoppers. Next is traps. So traps are used for the sample mobile insects. So basically there are two kinds of traps. One are the attractive traps and second are the passive traps. So attractive traps means they rely on the visual appearance or the chemical stimuli. So visual appearance means they were really attractive in color or shape, which attracts the insects. Or the chemical means in this chemical kind of thing they'll use the food source or the pheromones. So basically pheromones are the sex hormones which were released by the females to attract the male insects. So in the market they artificially synthesize the pheromones and they apply on the trap, so that it attracts the male insects, thinking of that their females is calling for mating, but no, it's a trap. So these are chemicals. So these attractive, with the help of the visual or the chemical stimuli, they lure the insect. Next are passive. So in this passive, the insect they get stuck accidentally. So this is the passive. Next is so you can see these are the examples of attractive traps, like the yellow sticky traps or this red kind of thing which is attractive, and the sticky kind of thing is applied on the surface of these traps, so that they get stuck on these traps. Next is attractant traps. So in these attractant traps, food source is used, like ammonia. Mostly ammonia is used, and in ammonia, ammonium acetate or ammonium carbonate is used. So in the picture you can see fly trap attractant is there and the fruit fly attractant. So these are the two food attractants which were used, like they are applied to the trap so that this food source they attract the insects so that they come in and they stuck in the trap. And these source they have a protein called casein in them, and these attractant traps they are oftenly combined with the sticky traps also. Next is pheromone traps. So I said pheromone traps, I mean the pheromones they are the synthetic version of the natural scents produced by insects to attract the males. So these are usually species specific, like for each and every species there were different kinds of pheromones. So, and also these synthetic pheromones they are available for many pests like cutting moth, cutworms, grapefruit borer, etc. So in the picture you can see there is a pheromone called gova moth pheromone trap. So for each and every species there is different kinds of pheromones produced artificially. Next is pan traps. So basically these traps are attractive in color. In case of color you can see the yellow is there, blue will be, and the red also will be. And the main advantage is they are inexpensive, and also they can last for several seasons with a good maintenance, and also they are easy to check. Since they are in this we'll use the pan kind of things, and we'll pour some, it can be water or chemicals, it can be insecticide, so that this color it attracts the insects, and these insects they slip into the water or the chemicals and they eventually die. But the disadvantage is when we are collecting these samples they may spill. This is one of the disadvantage. And second is when we do the irrigation to the crop, so if the water is fall, I mean, so if the water falls uh, into these pans, so the chemicals they get diluted. So this is one of the disadvantage. So example is blue trap for aphids. Next is pitfall trap. So this is the passive trap. So what are passive traps? Passive traps means these insects they will get accidentally trapped. So in the picture you can see in the ground we'll just dig a small hole and we'll put a can or a plastic kind of thing into the hole, and either we'll fill the can with water or the chemical, and we'll just rearrange the upper surface of the soil so that the insects they don't get suspicious. So the main advantage is they are inexpensive and they are easy to check and they can last for several seasons. But the disadvantage is the dirt in the samples. So this is one of the major disadvantage. And these pitfall traps are used for sample ground beetles and spiders. And the main another advantage is we can literally keep this thing like this. I mean, keep this can like this in the ground like for many seasons, as I've told you before. So no need to change it, and this is really cheap and it is used in all kinds of field. Like if you observe in different kinds of fields, you can see them. Next is shaking and beating. So this is the common kind of method of sampling. So basically under the plant you'll keep a white paper or a white cloth and you start shaking its branches. So the insects they'll get collected on the white paper. So basically we'll use helicoverpa. I mean like the helicoverpa is collected in this method of shaking and beating. Next is light trap. So this is the mostly used visual trap for sampling of the agricultural pest. So this is used to sample the moths, hoppers and beetles, and this is used to collect the rice stem borer, that is the Scripophaga and Scripophaga incerculus. So this Scripophaga it affects the rice at its brood emergence. So this pest it is used, I mean it is collected by using a light trap. And next is, since you have collected the pest and you have took it to the laboratory and you've got all the kinds of characteristics, so you should prepare a data sheet. So what are the information, or you can say what is the data sheet contain? So it contains what are the seasons and what is the crop and what is the variety of the crop and what is the stage, and what is the date of collection, and what is the weather condition, name of the farmer, soil condition and soil type, and is there any previous history of the soil sampling, or what is the previous crop, and is there any pest observed in the previous crop, and what are the diseases, weeds and rodents which are affecting the crop. So these are the basic information to be collected on the data sheet. So this is it guys with the today's lecture. So in today's lecture we have seen what are the pest survey and sampling and the sampling methods and pest monitoring. So if you have any doubts, just kindly go back to the lecture and listen again. So thank you for watching. If you have any queries, you can ask us on the Instagram page, or you can post in the comment section. And also you can download this PPT in the link given below. And if you like this video, kindly subscribe to our channel. So till then, stay tuned, take care, bye bye.